Okay, protein synthesis. As we've been saying, we know protein synthesis is divided up into transcription and translation, and we know that transcription occurs inside the nucleus at the DNA, at a specific part of the DNA that's called the gene. So notice we have our complementary base pairings all the way down the DNA. Our guanine is always with cytosine, adenine is always with thymine, and notice we don't have any uracil in DNA because uracil is only found in RNA. Now during gene activation, usually a cell signaler comes in. Could be something like a hormone or other chemical comes in and basically goes to something called a hormone responsive element, or a different part of the gene that unlocks the gene. And then what we have is an enzyme called RNA polymerase. We know polymer means many units of something, and RNA, we're going to make many units of RNA. So what RNA polymerase is going to help us do is actually going to help us build messenger RNA. So it goes to this initiator, this control segment, which then basically says that it should start reading the template strand and start reading the actual bases of the triplets. Now notice we have our triplets on the template strand, and in our DNA the other strand is called the coding strand, and the group of 3 base sequences were called complementary triplets, and they were always complementary to the actual triplets. So A was with T, T was with A, G was with C, and so forth. So the next thing that has to happen then is our RNA polymerase starts reading this and attracting nucleotides, and specifically RNA nucleotides. So it should attract an adenine to the thymine, but to the adenine, you might think, well, it should be thymine, but because we're building RNA and not DNA, that means that we know uracil is actually complementary to adenine. So again, notice here we temporarily hydrogen bond the bases, 2 bases. Notice these are our hydrogen bonds, which are easily formed and easily broken. And then, so when we temporarily bond our nitrogenous bases together, we then covalently bond the actual phosphates and the sugars that make up the rungs together. So this is a much stronger bond than that was, as we're forming our actual messenger RNA molecule. Now again, notice this was our first 3 base segment was our triplet, and what we formed from that 3 was our first codon. Our next triplet, so if this was our first triplet right here, then our next triplet is GGC. So that means our next codon would be CC, and then hopefully you see that since that's a C, this has to be a G, so here we see CCG. Okay, and then as we continue, TCG, so the next should be A with T, G with C, and C with G. Now notice we never have a thymine over here because this is RNA, whereas we never have uracil here because this is DNA. So we just have to understand what's complementary to each other. Well, guanine is always going to be complementary to cytosine. Adenine will always be complementary to either thymine and DNA or uracil and RNA. Okay, here we see that our messenger RNA actually left the nucleus to a nuclear pore. So transcription's over. Now we're going to start translation. Now, the first codon here was always AUG, which meant that our transfer RNA had to bring an amino acid to this, and it went to this light subunit. Okay, so that means if that was U, this had to be A. If that was A, this had to be U. If that was C, this had to be G. And we know this had to be methionine because something about bringing methionine to this small subunit actually causes the heavy subunit to surround it, and then we can start reading the rest of our messenger RNA. So this first step is actually called initiation. Now notice our next codon here, since our anticodon is reading GGC, then that meant that this codon had to be CCG. But what's happening is this was our initiator right here. Now we brought our second amino acid. So what's going to happen, we're going to have an enzymatic reaction where we're actually going to join these two. And remember, anytime we join something, we always go through a dehydration synthesis. We always remove water from it. Even though they're not showing that here, we know that's what happened. Okay, so we're going to form this covalent peptide bond here. Then this is going to actually release its first amino acid, and then this transfer RNA will then be free to go pick up the same one so it can be used again. Okay, so we saw the first transfer RNA that brought methionine, so that would be methionine. Here we saw that it was released. Now that same transfer RNA will go pick up more methionine to be used somewhere else. Here we see then our next amino acid was covalently bonded. That peptide bond is that covalent bond between the carboxyl group of one and the amino group of another. Here we actually have basically bonded the second amino acid to the third. Then this will detach and leave. Our next transfer RNA with its anticodon will bring the amino acid in. It will read the codon, and as long as it's complementary to the codon, the enzymatic reaction will hook up our third to our fourth. So we are building our growing polypeptide. Okay, eventually we hit what is called a stop codon that basically tells us to cut off the growing polypeptide, and now that polypeptide will either go into an endoplasmic reticulum if the ribosome is on the endoplasmic reticulum, and if that's the case, it'll go into an endoplasmic reticulum where it will pinch off into a vesicle and be transported to the Golgi apparatus, and at that point the Golgi apparatus will do further processing and packaging. If this happened to occur at a free ribosome, one that is actually floating around in cytoplasm, then most of the time it will be a completed polypeptide or a completed protein. Well, let's look and see what would have happened if this was the ribosome right here, which made this rough endoplasmic reticulum, and if the growing polypeptide was actually formed in the ribosome, it would go into the actual endoplasmic reticulum. Remember, this is one of our membrane-bound organelles. Therefore, we can form a vesicle. So imagine that growing polypeptide went into this area, then it was able to pinch off, and remember, my favorite motor protein would then grab this and start pulling it and walking across the cytoskeleton to where it can fuse this membrane-bound vesicle with the growing polypeptide into it, or with the polypeptide, and actually fuse it with our Golgi apparatus, which is this structure, because our Golgi apparatus actually has enzymes in it for further processing and packaging of our developing protein. Okay, once we are in here, again, we see that membrane can bond to membrane and deliver substances. Now we can actually go through the different actually bodies of the Golgi apparatus and go from one area to another, where we can then, due to the enzymes that are inside, change our product into what we want it to become. And then eventually, according to what product we're making, whether that product is protein-based from the rough, or whether that product would be a carbohydrate or lipid-based from the smooth endoplasmic reticulum, what we would eventually want to do is either release the product through exocytosis, or we may even be making a product that we're going to embed into that membrane, because remember, our cell membrane is also a phospholipid bilayer, and it's ever changing, and sometimes we need to embed new phospholipids or proteins inside that, because remember, one of our main subjects of anatomy and physiology is understanding that cells are very dynamic, and we've all the time got to be changing and putting in different proteins as they degrade or as we need them in different situations. Last but not least, we can actually form structures for the cell itself. So one of our other organelles is basically lysosome, which pretty much has hydrolytic enzymes inside of it to do many cellular functions. So you can see that cells need proteins, because proteins do everything for the cells, and cells need to use both the ribosomes and to make the proteins. They need to use the endoplasmic reticulum to further package the proteins so they can send it to the golgi apparatus, where we actually enzymatically alter it and make our final or almost final product. Those products basically either will be secreted somewhere else through exocytosis, those products will be incorporated into the cell or into the cell membrane, or those products will be incorporated into the cell as a structure to help whatever the cell is do the type of job that it needs to do.